Welcome to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from across the schools and departments of the University of Cambridge, reflecting the pioneering work and diverse interests of members of Cambridge Neuroscience. Cambridge Neuroscience is currently going through quite a detailed consultation process to develop 6 new themes for the research we do here. Each of the next 12 talks will come from one of the 6 new themes, 2 from each. For more info on the themes and the talks covered in this series, please follow the links below and follow us on Twitter at CamNeuro. Welcome everybody to the last in the current series of the Cambridge Neuroscience Interdisciplinary Seminar Series. And so today it gives me great pleasure to introduce you to Professor Giovanna Mallucci. Giovanna is the Geest Professor of Clinical Neurosciences here at the University of Cambridge and is also the Centre Director of the UK Dementia Research Institute. So following undergraduate research in physiological sciences and medicine at Oxford, then going on to do her clinical training at UCL before a PhD. She's finally decided to join us here in Cambridge, and we're very lucky to have her. Her lab is broadly interested in the treatment of dementia, but specifically in the role of the unfolded protein in neurodegenerative disease, and of course, its pharmacological manipulation. So welcome, Giovanna, and we're all delighted and looking forward to hearing your talk.
Okay, right, I'll kick off. I'm sorry for the delay, everybody. I wish I could see you all, but I can't. But nonetheless, I'm very glad to know your names are there, and I'm delighted to be talking to you today. I'm going to give a very broad overview of the themes that interest us in the lab. I've called it rather grandly mechanisms to medicines, but mainly to emphasize the translational slant in the programs that we've been pursuing. So we're broadly interested in neurodegenerative disease, which of course are a very wide-ranging group of disorders, going from the very common to the very rare. I'm just going to get a pointer here. I think most people will know someone with Alzheimer's disease, whereas very few of us will know the very rare prion disorders, but they all have very similar shared principles, these diseases. And collectively, they are a huge global and societal problem, and rapidly becoming the commonest cause of death in the developed world and also in the developing world as it ages. But traditionally, a lot of focus has been on each disease from its very much a pathological perspective, from its own specific misfolded protein and its own specific pattern of neuronal loss. But in fact, there are some very common processes that dominate the picture for all of these disorders. One is an inherent decline in synapse number over and above what happens with normal aging. And the other is the accumulation of misfolded protein. And through both through indirect and direct effects, these combine to lead to neuronal loss, which of course is a feature that affects all of them. Now, this slide is important because it says so much about the redundancy of the system, the short window we have to treat neurodegeneration. So really, when you've lost about 60% of your synapses, you then present in clinic with your early memory loss or your early movement disorder or whatever the feature of a particular disorder is. But then there's only a very narrow window from which you lose further synapses, you lose all the trophic support to neurons that is so essential. So then you're into the irreversible stage of neuronal loss, which is institutionalization and death ultimately. So this narrow window is the sort of reversible, tractable phase of neurodegenerative disease that we are so interested in. And both of these processes contribute to shaping this window and this onset, very much determined by the slope of this curve. Change the slope of this curve and you change the onset of neuronal loss and the mechanism, the outcome of neurodegeneration. And of course, common mechanisms may give us common cures. So rather than having a disease-specific approach, we're going for a sort of mechanism-driven approach. So when it comes to studying these mechanisms, I gave you a list of disorders, of which the rarest was prion disease. However, all the other disorders, the mouse models have really quite marked limitations. They often have a very partial phenocopy of the disorder. So many of the Alzheimer's models model a bit of amyloid deposition or a bit of tau phosphorylation, but really they are not, those mice don't have Alzheimer's disease. But these mice do have prion disease. This is biochemically, pathologically, phenotypically, and clinically prion disease in a mouse, which completely recapitulates the human situation. And above all, what these mouse allow us is to study the relationship between aggregate formation, and these are the prion protein aggregates or amyloid, and the loss of brain cells, which here you can see a degenerated hippocampus in that mouse. This is CA1 to 4. And you can, this is a healthy dentate gyrus, this is a degenerated CA1 ribbon, CA1 to 4 ribbon. So the sort of fundamental principle underlying all of these disorders is protein misfolding. And here there's a cartoon depicting the misfolding of the prion protein, which here you have in blue circles, alpha helix rich, protease sensitive native form, which is converted to an aggregated beta amyloid aggregated form during disease. And there's been a lot of interest, of course, in this form, and I'm sure you'll all be aware because of its transmissibility and infectivity, and it's the cause of mad cow disease and human Creutzfeldt-Jakob disease and the new variant of that. But the bottom line is that this protein misfolding is occurring in cells continuously and does represent a stress. And just for the non-biochemists, I like this image of protein misfolding. If you think of this deck chair as a protein correctly folded, and these as misfolded proteins, you can see very quickly how the same primary structure assumes a completely different tertiary structure and becomes non-functional and stacks up. And in response to this stress, and this is highly simplistic, obviously, representation, all cells engage an unfolded protein response. And this is a complex, well-orchestrated response of both transcriptional and translational elements. And the transcriptional side lead to stress-related genes, chaperones, lipid synthesis, increasing endoplasmic reticulum capacity. But the one branch that we're particularly interested here is the translational response, where PERK, which spans the endoplasmic reticulum membrane, is activated when misfolded proteins accumulate within the lumen of the ER. And this is highly oversimplified, but basically accumulation of misfolded proteins is sensed by a sensor called BIP, which dissociates from PERK. PERK dimerizes autophosphorylates, and that activation of PERK puts a phosphate group on this molecule, which is called EIF2 alpha, and that blocks translation at the stage of initiation. So you can't put methionine on the beginning of a polypeptide chain when this is phosphorylated. So this is a transient response usually, reduces translation. The other arms of the UPR come in. There's protein, there's new, there's transcription of new stress-related proteins that are made, and lipids, etc. And this is usually rapidly reversed and dephosphorylated by specific phosphatase of the IF2 alpha, which is GAD34, and that restores translation. So this is a classic homeostatic mechanism that usually responds rapidly to a stress and gradually and quickly resolves. What we discovered in prion protein, in prion disease, in our prion disease mice, was that as this misfolded protein accumulates, here is the levels of prion protein rise, there's a very sustained and increased level in the levels of EIF2 alpha, paralleling that accumulation. And interestingly enough, this early increase in phosphorylated 2 alpha is first reduced, but then that fails. So we have a failure of the dephosphorylation, which allows its sustained elevation for weeks rather than minutes. And you can imagine that that has fairly marked consequences. And one of those is a very marked sustained reduction in global protein synthesis rates when 2 alpha is phosphorylated for such long periods of time. And this is global protein synthesis, but one of the things we noticed that was that amongst the proteins that are reduced are synaptic proteins, and any particular pre or post synaptic protein that you look at has very marked reduction at the time of 2-alpha, high levels of 2-alpha phosphorylation. And this is really a critical point, because the synaptic proteins, of course, are needed for synaptic structure, maintenance, and as we said, for that trophic support to neurons. And there's immediate behavioral change and memory loss. This is a mouse that's performing a motivational task of the hippocampus called burrowing that is you can measure, and that's reduced. Memory is also impaired, and neuronal death follows quickly after. And here you can see these spongiform vacuoles, but then the degeneration of these neurons. So our focus and interest in common mechanisms meant that we put forward the hypothesis that actually this sustained activation of this branch of the unfolded protein response was contributing to neurodegeneration indirectly through a deprivation of critical protein synthesis rates. And to prove this, we did a genetic proof of principle experiment to try and avoid the phosphorylation of 2-alpha during the evolution of disease. So we did 2 interventions, RNAi of PRP to stop the buildup and stop activation of this pathway. We put in extra GAD34 into neurons in the hippocampus. These are all delivered by stereotactic viral injection. And we use salubrinol, which is in some way prevents the dephosphorylation of 2-alpha. So if you look at here at the conditions, if you look at the GAD34, the RNAi and the GAD34 in particular, here is the RNAi of PRP the GAD34 prevents dephosphorylates the high levels of 2-alpha, whereas the RNAi prevents the high levels of phosphorylation, and salubrinol does the opposite. So biochemically, all these interventions are working as designed, and the result of that is the restoration of protein synthesis rates in the hippocampus, and the further reduction in salubrinol. And this is profoundly neuroprotective. So these mice all have the same stage of prion infection, the same level of prion amyloid in their brains, but where here you have this marked neurodegeneration, where you've dephosphorylated 2-alpha, you've got marked neuroprotection. And this is reflected also in a really quite significant increase in survival, and this is just by focal hippocampal injection. So we then propose that actually, if you can restore translation by reducing the phosphorylation of 2-alpha in these disease contexts, you will have neuroprotection. And this is interesting, this is not just, these experiments were all done with viruses targeted to neurons, but it's just as strong an effect if you target the virus to astrocytes. So here we're using an astrocytic promoter, and you can see that this neuronal ribbon is as protected if you target the UPR in astrocytes, or you target 2-alpha phosphorylation in astrocytes. And this is thought to be through, or we've shown that it's when an astrocyte has PERK activation, its secretome changes, and there's a reduction in neurotropic factors in extracellular matrix compartments that are essential for synaptic support, and this is reversed by targeting the astrocytes. So there's a non-cell autonomous effect, and that was genetic proof of principle, which affects both cell autonomous and non-cell autonomous through the restoration of protein synthesis rates and genetically via GAD34. So can we do it with drugs? So when we were first doing the original study with the genetic rescue in neurons, we heard that GSK had made this very, very specific inhibitor of PERK as an anti-cancer drug, but it wasn't very successful as an anti-cancer treatment. So in any case, we asked them if we could use it, and we performed an experiment where we gave the drug orally. We established that it got into the brain, we gave it orally, and we gave it at 2 time points which were clinically relevant in an adult, in a human disease paradigm. So this is early synapse loss, which would be equivalent to an early memory loss. And also later, when you've got decline in protein synthesis rates already, early spongiosis and behavioral deficits. And I'll just show you some videos. These mice have all got the same stage of prion disease. This one is treated, it's burrowing effectively. This is a CA1 dependent task. And this mice is given vehicle mouse, and it's not behaving in the correct motivational manner. As disease progresses, you can see, again, littermate, same stage of disease. Vehicle treated mice has hind limb paralysis, whereas this littermate is absolutely fine. And again, as disease progresses, this mouse is akinetic, it's ataxic, it's got this dystonic posture, it nearly falls over there, and there's its littermate. So this is and finally, just a terminal disease, you cannot put a healthy mouse on its back, but this is so ataxic and so profoundly uncoordinated and neurologically damaged that it can't right itself. So this is clinical cure. This is the first small molecule to actually cure a neurodegenerative disease. And it was doing it through the restoration of protein synthesis rates, and all the signaling downstream in the pathway is also affected in that direction. And you can see really how profoundly neuroprotective this was. But if you compare the hippocampal rhythms in treated and untreated mouse, and remember that the amyloid load and the misfolded protein load upstream is equal in these two animals. And it was hailed that when this paper came out, there was a big news storm, and it was hailed as Alzheimer's breakthrough, as a turning point in treatment. And we were really wondering how the general press had come to this conclusion. But of course, they're well informed, and actually they were aware that the PERK branch of this disorder is overactive in a region-specific way in many neurodegenerative disorders, including Alzheimer's and Parkinson's, frontotemporal dementia, progressive supranuclear palsy, and also ALS. And in fact, a number of papers came out. In fact, that when we published the small molecule data that week, Eric Klan from NYU, from Columbia, published another experiment with genetic suppression of other eIF2 alpha kinases, alleviating memory deficits in Alzheimer's mouse models. Nancy Bonino tried the same molecule in an ALS fly model, and Claudio Hetz in a Parkinson's model. So that, and then we also found a dementia model itself, a tauopathy model, where treatment prevented or was really quite markedly neuroprotective. So we've sort of proved our point that if you act downstream of the misfolded protein targeting a common mechanism, you can provide neuroprotection irrespective of the particular neurotoxic protein species, as it's called. But there was a but, and the but was, as I think had probably been predicted by the people in the field considering PERK inhibitions, there was pancreatic toxicity of this molecule. So here's the pancreas, which is essentially, it's secretory tissue, so it's really loaded with endoplasmic reticulum, and it absolutely needs a functioning unfolded protein response and all branches to be functional. If they're not, this is necrosis of, or apoptosis actually, sorry, of acinar tissue. So this is the secretory tissue just dying when you inhibit PERK as effectively as GSK does. So we turned in the press. What I haven't mentioned is that protein synthesis, in particular to alpha phosphorylation, is really essential for memory formation. So where you have high levels of phosphorylated 2-alpha at synapses, LTP is impaired and memory formation is impaired. And Naum Sonnenberg and Peter, working on the control of memory and cognition through 2-alpha phosphorylation levels, Peter and Carmela Sidorowski made this molecule, which is called ISRIB. A great deal is known about this now, but this is 2013, and they made ISRIB, and they showed that this molecule enhanced cognitive memory in wild-type mice. So we made ISRIB, which acts downstream of GSK. It acts, we've got David Ron in the audience who's defined ISRIB's exact point of binding on 2-alpha's interactory IF2B. So it's very well known where ISRIB acts. But bottom line is it acts downstream of PERK. And when you give it to the prion mice, It's as neuroprotective, but it's not toxic to the pancreas. And this is because and this is a publication that we did with David Ron and his team, where we show that the effect of ISRIB on this signaling is partial rather than complete. The GSK is almost complete inhibition, whereas this partial restoration. It allows enough room for the brain to make the proteins it needs to survive and the pancreas to switch off to a degree that it can survive. So although this was a much smaller story, as it were, it's very important in terms of validating this whole approach. You know, if you can turn it down rather than off, then this pathway does become a viable target for therapy, and not just in neurodegeneration, quite a wide spectrum of disorders. So this indeed has become a wide focus for drug discovery. So while, and there's a great deal of drug discovery, and ISRIB has been in phase 1 clinical trials, and there's new ISRIBs, and there's lots of other interest in similar molecules and developing similar molecules. But whilst in the lab, I had a really fantastic PhD student called Mark Halliday, who instead of making, took a repurposing approach, and instead of trying to design new drugs, he screened a library, an INS2 custom library for activity in this branch. And this is a cell line given us by David Ron, where you can measure signaling through the PERK pathway by measuring luciferase activity driven by the activity of the CHOP promoter. And this will only be driven by when 2-alpha is phosphorylated. And you can see here the change in luciferase expression with the ER stressor capsigargin. This is the almost complete inhibition by GSK. Here's ISRIB. And here are 2 molecules in his library that have similar activity to ISRIB in this screen. And long story short, we put these drugs into prion mice, and they are as effective as the GSK compound or as ISRIB in neuroprotection. And this one is a licensed, safe drug that's widely used in the elderly and in human populations. So these 2, DBM isn't licensed yet, but it has been in phase 2 trials for cancer. But again, it's further along the pipeline, as it were. And trazodone is a dirty drug that's been around for a long time, but it does work in this capacity. And this is some unpublished data that showing you a technique called FunCat. Here we're using a non-canonical methionine called AHA, which can be, through click chemistry, can be bound to fluorophore, GFP in this case. And you can see where AHA is incorporated, all new proteins are labeled green, basically. So this is all new protein synthesis in a hippocampal slice in a control mouse brain. This is the protein synthesis in prion disease at about 8 weeks. So you can see 9 weeks, but quite a really, especially in the neuropil. This part is synapses and dendrites and you know, what we call neuropil, a lot of the processes of the neurons, and these are the cell bodies, but the really marked reduction in protein synthesis rates. And 2 weeks of trazodone treatment lights that back up again, as you can see, particularly again in the neuropil. So a real effect on restoration of protein synthesis rates. And this is again unpublished data where these are normal control animals, these are prion diseased animals, and this is trazodone, and this is a heat map, and I won't go into it, but there's a lot of synaptic proteins and other specific proteins that are in these that are specifically recovered by trazodone treatment. So translating this into patients, into human studies, we've found a way of imaging protein synthesis rates in humans using a similar methods to what I've just shown you, really. But here we're using a carbon-11 labeled leucine, which is an established method measuring incorporation into proteins in living patients. And this is preliminary data. We've got more data now, but I haven't got more scans to show you. But this is the mean of 5 control. So we've got elderly patients, we've got people with early Alzheimer's disease, and people with and their age-matched controls. And this is the signal throughout the brain of newly incorporated protein synthesis during the duration of a PET scan. That's in 5 controls, and I've only got one patient to show you, but I think you can appreciate the change in signal. This, of course, needs confirming, and we have more data, but we need to quantify it and measure it accurately. But this was certainly very suggestive. And interestingly, if you do these what are called T-maps, the main brunt of this difference is in the hippocampus and entorhinal cortex, which is the area of interest. And of course, our question is, can ziprasidone restore protein synthesis rates in human brains, and could that bring cognitive benefits? So we're hoping to do clinical trials in dementia. We have a protocol. We have we're going through various rounds of trying to get funding, but we're not there yet. But that's the next step for this. Oh, sorry, that's my watch talking to me. So if I have a few minutes left, I think I've got 10 minutes left. I just wanted to just quickly talk about the other side of interest in our lab, which is in the processes driving the decline in synapses, which A, happen with age, and B, are so accelerated in disease and are so critical to making us vulnerable to all these other stresses that are going wrong, these other dysregulated homeostatic processes. Just to remind you, this is a neuroscience seminar, but anyway, what a synapse looks like and what it looks like on electron microscopy. A classic synapse will have a density, a cleft, and presynaptic vesicles. And I'm going to show you more of those. But the thing, as I've said many times already today, loss of synapse occurs early in neurodegeneration. Here in red is the point at which we get UPR activation and then neuronal loss. So you can see that this early loss of synapse sets the scene for vulnerability. And why are our synapses so low at this stage in disease? Well, the phenomenon of structural plasticity is a balance between pruning and regeneration, which goes on all the time in our brains. Obviously, during development, regeneration or generation is obviously very florid, and we know that pruning increases with age and in disease. But certainly, the balance between this capacity will affect net synapse number. But how do we test the capacity for structural plasticity? Now, you can put a window onto in the cortex, in the cranium of a behaving mouse, and with very sophisticated imagery, watch synaptic boutons and dendrites come and go. But that's very high intensity and specialist. So we did a different thing. We turned to biology. We turned to the phenomenon of hibernation. So hibernation is well known to all of us as a seasonal adaptation to cold, but actually to lack of food. And what is involved is a lowering of body temperature, a reduction in metabolism, a massive reduction in protein synthesis rates. And one of the phenomena that is observed, if you do an electroencephalogram on a hibernating animal, that's virtually flat, and that's because they actually dismantle their synapses during hibernation. So this is a well-known fact. So the whole neuronal arbors retract, and synapses are disassembled, as it were. So here is the control synapse of the PSD. It's gone on cooling. So that means that transmission can't occur. So the vesicles are pooled, and then it's restored and rewarming. The assumption that we made, or the hypothesis that we made, was that this is almost certainly that hibernation is probably an extreme phenomenon of something that occurs more generally and represents a sort of dismantling and reformation or regeneration of synapses. And we asked ourselves if this capacity is actually not just for hibernators, but if this is actually can occur in wild type mice. So we tested the capacity of wild type mice for structural plasticity. Here I've pseudo-colored the synapses for ease of looking at these micrographs. And we cool them to 16 to 18 degrees, which is the core temperature of a small hibernator. We use a biomolecule called 5'-AMP, and that rapidly and safely induces torpor, and then we keep them cooled for a few hours, and then we let them gradually rewarm. And what you can see is that synapses, just as in real hibernators in a laboratory mouse, are reduced and then reformed on cooling and rewarming. So this was exciting, because although this is just our control, this actually shows that this phenomenon is preserved. It's preserved in non-hibernating mammals. And then we moved on to ask about structural plasticity in disease models. So now we can turn to our prime mice, but also to other mouse models where we do, this is an Alzheimer's mouse model, where synapses are indeed lost. And we need to test the capacity for plasticity before synapses are lost. So we have to test them very, very early in disease. And what we do with these mouse models is exactly the same. We cool and rewarm them and count their synapses. So at 4 weeks, in the prime course, they die at 12. They net loss of synapses at 7. They are still plastic, but at 6 weeks, which is they have indeed lost their capacity for synaptic plasticity, for structural plasticity. And this is reiterated in the Alzheimer's mice, where they have net loss of synapses at 4 months. 2 months, they're plastic, as you can see, and then they've lost that plasticity at 3 months. This therefore precedes synapse loss, and it's a very early deficit in what we call the regenerative capacity or the restorative capacity of synapses that precedes everything else. And what is the mechanism? Well, we know that cooling and hibernation are neuroprotective, and we also know that they engender a cold shock response, which, unlike the heat shock response, involves the upregulation of specific cold shock proteins, which are RNA chaperones rather than protein chaperones, as well as a global reduction in protein synthesis, except for these particular cold shock proteins, one of which is called RBM3, RNA binding motif 3. It's highly expressed in brain, and here you can see its induction, and this is known to increase on cooling in bears and ground squirrels that hibernate, and even humans sleep, it varies. But here it's the induction on cooling in our mice, and in both the prion and the Alzheimer's mice, that induction on cooling fails at the same time that the capacity for structural plasticity fails. So clearly, it may well be associated with plasticity. How do we show that? If we increase the RBM3 levels in these disease models, can we restore synapse regeneration? So we did this in 2 ways. First, we did it just by cooling. So we take prion mice, we cool them early twice while they have the plastic response, and then we let them proceed. And what that does is gives you a very high sustained level for several weeks, which makes sense because of the hibernation duration, the bouts of torpor, over these high levels of RBM3. And the effects of that, here's a control uncooled mice at 7 weeks. It's synapse, mean synapse count, representative synapse number. And these mice have been cooled, and they don't have synapse loss. So they have completely protected levels as wild type of synapse degeneration through having been cooled twice, despite having established prion disease and established prion replication. And this restores their behavioral deficits, their memory, and again, it's highly neuroprotective. And here we've done nothing except cool them. We haven't put in any viruses or genetic modulation or small molecule. And that also increases survival very significantly. Here it's the outlier, which tells you really quite how protective this can be. Conversely, RNAi of the RBM3 prevents the protective effects of cooling. So cooling is messy, obviously, and does much more than increase RBM3. So if we just put in RBM3 alone, so this is now like the GAD34 experiment, but we're putting in RBM3 and RNAi, Here's the controls. We've done it in both prion and Alzheimer's mice. And at the equivalent time point, you can see how beautifully the synapses are protected. RNAi reduces it. This restores transmission. Again, behavioral deficits and memory. And again, it's beautifully neuroprotective. So this is a second way of targeting a common process that can allow protection in multiple disease models. And it also increases survival. So we have this very brief, very sort of simplistic view, which cooling induces RBM3, induces neuroprotection. What we really want to do is induce RBM3 without cooling, because cooling is widely used therapeutically, and I'll talk about that in a second, but it's really unpleasant. You get pneumonia, you get blood clots. You can't do it for very long. So if we could pharmacologically induce RBM3, that would be an interesting approach. Again, the BBC came to our aid, and in fact, we'd cooled prion mice to 16 degrees. So I really wondered how the media might get at dementia therapy through this. But in fact, they've shown us a picture of bears. Bears are large mammals like humans, and bears hibernate at 35 degrees, which is what we use therapeutically in hypothermia. What's the relevance for human disease? Is this modulatable? We do know much more about this pathway. We know that RTN3 is a major mediator of that. There's a reticulin protein, there's an ER shaping protein, and we know that this is mediated upstream through a non-canonical track B mediated signaling cascade, and that is druggable. But here's two examples of hypothermia. This is therapeutic. This is a cooled baby with hypoxic ischemic encephalopathy. They're cooled 35 degrees. And these are people who are voluntarily hypothermic. They're cold water swimmers. So we worked with these people. We worked with both the babies and these people. And here's the baby's RBM3 levels after cooling. So this does happen both in blood and in brain. And here's the swimmers. So we worked with a bunch of swimmers where we let them get hypothermic, because you obviously can't get permission to cool people to hypothermia for no good reason. But these guys were already hypothermic, and they also have RBM3 in their bloods. And only the swimmers have the raised RBM3, and no non-swimmers did. So, but of course, as in any human population, not everybody has it. So I'm going to stop there. There's much more to say. We also know we've detected a channel sensor for temperature that also couples and raises RBM3 and is druggable. So there are lots of ways through, but really what the point I want to make is you can approach these disorders not in a disease-specific, homeostatic processes that impinge, and there are more of them. There's autophagy, there's mitophagy, there's oxidative stress, etc. But anything that can change the shape of this curve would defer neuronal loss, which would really change the face of these disorders and their clinical manifestation really quite dramatically and meaningfully. And I still do memory clinics. I'm a neurologist. If I could hold my patients when they present with their early memory loss and stop that deterioration, it would make an enormous impact. And in fact, one of science's unanswered questions, a 5 to 10 year delay in this late onset disease would improve old age for millions. So I need to thank my group, who have been incredibly helpful. These members past and present have all contributed to these two stories, my funders, and my collaborators, and this is me and several of my team in the Baltic having a bit of coal shock at a recent meeting, and I need to thank my collaborators as well, who've been so important in getting all these papers. I know David's here, the clinical team that have helped, Anne Willis and Martin were instrumental to all the UPR and also the cold shock work, and Peter Fisher, and others, many others. Thank you very much. I'm going to leave that one up.
Thank you, Giovanna, for that wonderful talk and insight into your research on neurodegeneration and your fascinating work on cold water swimming. Maybe something for us all to try out in the new year. So that's it for this term. I'd like to take this opportunity to thank all of our speakers this term. Make sure you catch up with all the seminars that you've missed on this YouTube channel. We'll be back in January when we will welcome Professor Sarah Baker on January 18th. Sarah is a professor at the Faculty of Education here in Cambridge, and she will share her research on identifying effective interventions in early childhood education. Sarah is our first speaker in a fabulous lineup that we have planned for 2022. For more info on this seminar series and all things neuro related in Cambridge, follow us on Twitter at CamNeuro and follow the links below. See you next time.